All right. So essentially the solution that we found, that there are proteins in the brain, they actually clump within the synapse. And when they clump within the synapse, they change the synapse, they change the activity of the synapse, the way the synapse communicating with each other, and helping to create memory, store memory. That's what we found.
So the clumping is good?
Exactly. So then we asked, okay, the protein is clumping. What sort of protein clump is this? And we looked at them very carefully, and we surprisingly found that these clumps looks like protein clumps that we have been studying for a long time as the cause of many of the diseases, that is amyloids. So what we started out to understand how we create memory and how we, animal hold on to them.
Yeah.
And it turned out that at the very bottom of that process is a protein that clumps and create a stable substrate or representation of memory. But the protein clump looks very much like proteins clump we've been associated with loss of neuron, loss of memory, various diseases of the human nervous system.
These are what we call in the brain the tangles and
exactly. Tangles and the plaques.
Tangles and plaque, yeah. So you're saying that the tangles and plaque are made up of the same substances as the memory connection in the synapse?
No. Two types of protein. There is one type of protein that is helping us to create a memory, and they clump. But there are other proteins that are also can clump. But when these other proteins that are involved in, suppose, Alzheimer, Parkinson, these are different proteins actually. For different diseases, there are different proteins.
Okay.
And when these proteins clumps, they cause disease. But there is a different protein, when it clumps, it causes memory stabilization, memory formation. But if you look at the clump, how do they look, how they organize, they look very similar.
Okay.
And now question is, what's the importance of the similarity?
Yeah.
The importance is that the basic idea for all the diseases is that the reason that different proteins causing you all this problem, when they clump, they form a very specific type of structure. These structures are so stable, they form like a fiber, like a string.
Yeah.
And once they form the string, the cells don't know what to do with them, and they causes all these bad things and eventually leads to loss of memory, loss of synapse, loss of neuron, and bad things. And most of the diseases are actually this nature, how the strings are basic properties. And they have
of the degenerative nervous diseases.
Nervous diseases.
Yeah.
What we found that a different protein can form exactly same type of string, can be organized exactly the same way. And you would predict that if they're organized this way, this would cause disease, but it doesn't. It actually does something exactly opposite. And so
that's an important point. Say that again.
Yes. So the thing is that, if some proteins, when they will sort of organize themselves in a string, in a fiber, they will lead to bad things.
Yeah.
And a different protein that we studied, it seems to be forming a very similar type of strings. It's also organized exactly the same way. And normally you would predict, if you do not know anything about it, this protein should also cause bad things because it looks very much similar.
Yeah.
But it does not. It does exactly opposite.
So I would think the challenge then would be to figure out how to dissolve the protein that make up the tangles and plaque without dissolving the important ones that are sustaining memory.
Exactly. Very good. So I think I succeeded in explaining the basic problem.
Very
good.
It
immediately raises some very interesting, questions. And I think as a scientist, that's the part I'm most excited about.
Yes.
So if they are both looks very similar, they're organized. So why is bad and why the other one is good? Is there any way they're different? is there anything that is different we haven't been able to pick it up? That's question number one.
Yeah. So if you could find something that was different, then you might be able to target it.
Exactly.
Yeah.
And now we have some hint because we have solved so the atomic level, we know how they're actually built, and we have some hint. So we will
at the atomic level.
Exactly.
Wow.
Because that's the way you want to go.
Yeah.
The second thing is even more intriguing. That is the way we thought about so far, that how these strings or fibers are formed in the brain and in many other places, that these are uncontrolled because the body did not want it. It happened because something went wrong. Okay,
Yeah.
It's unintended. But what we are finding, our study would suggest that there are cases where brain actually intentionally building these fibers. Okay, so if they're intentionally building them, that means they also know how to take it apart.